Hi, everybody. Um, Welcome to today's class on kidneys and excretion. Uh, I would ordinarily start class with a would you rather, so I may as well go ahead and start with today's would you rather, although of course it's a little bit more difficult to play at home. But very quickly, the would you rather for today is would you rather be born with an elephant trunk or a giraffe neck? And again, I don't know that there's particularly a right answer there, maybe personal preference, but I think I'd have to go with a giraffe neck. So we're going to do a couple things today. We're going to talk about the basic structure of a kidney and how it works as an excretory organ. In other words, it's an organ which helps to excrete substances out of the body, specifically waste products. But we're also going to sort of briefly mention the fact that because we just recently talked about the endocrine system, that the kidney and kidney function is to a large extent under the control of different hormones that can control the amount of urine and the type of urine that's produced. There's a great YouTube video here. Hank in the crash course does an amazing job of describing the kidneys. So you may want to also watch the crash course videos in addition to this, or perhaps even in place of this, but I'm certainly going to cover the things that I think are the most important in terms of understanding the kidney and kidney function. If you wanted to get the extra credit on the next test, the Nerdy by Nature video is the TED Ed talk entitled, Is It Bad to Hold Your Pee? Spoiler alert, the answer is yes. And if you wanted to figure out why and what happens if you hold your pee too long, please watch the video. Again, it's only about 4 or 5 minutes out of your life, and it's probably, I think, an interesting watch and should help you gain the extra credit point on the next test. So let's just jump right in, and I wanted to give you sort of a quick overview of the kidneys and what the kidneys actually do before we sort of dive more deeply into the specific anatomy physiology of the kidney. Now, the kidneys are, of course, kidney bean-shaped organs, which are located in your abdominal cavity, but fairly deep in. They are located below the ribs and the diaphragm. And if we did the dissection of the rat, which we obviously didn't get a chance to do this year, or if you look at some anatomy online, you will see that the kidneys are located fairly deep in behind all of the digestive system organs. You would actually have to remove or certainly move around all of the digestive system organs to get at the kidneys, which are located just sort of almost buried in the muscles in your back. They're actually a little bit sometimes hard to see because they're covered and held in place by some connective tissue and mesentery, but they're located sort of, at the middle of your abdominal cavity below your ribs and diaphragm. Now, the kidneys are what we think primarily as being excretory organs. In other words, organs that help to remove waste and waste products out of our body. And certainly that is one of the functions of the kidney. But this is another interesting function of the kidney, which is to maintain osmotic balance, or the water content of the blood plasma in our bodies. You may remember in our last lecture on the endocrine system that we have an endocrine system that helps to maintain a whole lot of things within a fairly narrow range of values, right? So we have a very narrow range of values in which our body temperature can be can operate, or our bodies can operate in. And so it also is with the levels of water in our body. We don't want our blood plasma to be too hypertonic or hypotonic. We need to keep it at a fairly constant level. And so the kidneys are actually primarily osmotic organs. They actually help to maintain osmotic balance as much as they do excrete waste products. So here's another way of sort of thinking about this. This is the basic structure, again, of our kidneys. We have, you'll see perhaps these two big blood vessels here, which you probably could identify if you thought about them. One is the aorta, and the other is the inferior vena cava. And so blood leaving the aorta actually branches off, some of that blood branches off and enters the kidney through these arteries, and they're called renal arteries, R-E-N-A-L, renal means kidney. blood then enters through the kidney. It's filtered in these little microscopic structures called nephrons. So fluid is actually pushed or squeezed out of the blood and into these collecting structures, filtering structures called nephrons. And any blood which or liquid which hasn't been squeezed through returns through the renal veins into the inferior vena cava and back up to the heart. So it's been filtered at this point. Now, on an average 24-hour day, the kidneys may actually filter 180 liters of your blood. In other words, 180 liters comes into the kidneys, but only about 1.5 liters of urine are actually produced through the kidneys, right, and empty into the bladder. So in other words, you only produce about 1.5 liters of urine every day. So if you want to do the math here, you only make about a liter and a half of urine out of 180 liters of fluid which is pushed through the kidney. That means that that fluid has to be going somewhere. And in fact, the majority of the fluid which is actually pushed into the kidney, into those nephrons, most of that is reclaimed back into the body. So 99.99% of the fluid that moves through the kidney every day is reabsorbed. That means that the primary function of the kidney is actually maintaining water levels in the body. And a second function is to excrete some of the waste products of metabolism. And so let's quickly take a look at the waste products of metabolism that are excreted in the water that actually does filter through and become urine. And again, without dredging up too many horrible memories of our biochemistry unit way back in probably August or September of last year, you may remember that the building blocks of proteins are things called amino acids. Now, here's a typical amino acid. It has a carbon molecule in the center with a branch or side group called a carboxyl group. They all have a hydrogen atom. All the different amino acids have other things attached to this central carbon. They call it an R group. This is the part that varies from one amino acid to the other. But here's the part we're going to pay attention to. All amino acids have an NH group attached to them. And as we metabolize or break down amino acids into smaller and smaller bits, the amino acid groups are cleaved off. And so what we're left with is NH2, which becomes NH3. And you may remember from chemistry last year, NH3 is ammonia. Ammonia certainly isn't something that we want in our bodies, and yet it is something that is the product of protein metabolism. So the NH3 groups is actually, in mammals at least, and in some amphibians and sharks and bony fishes, but certainly in us, the NH3 group is metabolized in our livers into a slightly less toxic substance called urea. Birds and some insects and reptiles actually go a step further and make something called uric acid, whereas most fish excrete pure ammonia. Bottom line, if you look at these little blue ends here, these are the nitrogen groups that used to be on proteins, because all of these animals metabolize proteins, and all of them wind up with that NH3 group as a waste product that has to be excreted. We just happen to excrete it as urea, which dissolves in our blood and eventually gets filtered through the kidneys and out through our urine. A very quick overview. In a few minutes, we're going to really get into the kidney itself. We're going to take a cross section of the kidney and look at it in a little more detail. But the entire urinary system is actually relatively simple in terms of the organs that make it up. We, of course, have a pair of kidneys on both sides of our bodies. What's not shown in this slide, but will be in the next slide, is the renal artery and renal veins which feed the kidneys. And then we have these big, long collecting ducts or duct-like structures, long tubes called ureters. So eventually the substances that are filtered out of the kidney wind up flowing down the ureter into the bladder. Now the bladder has a muscular wall which can expand and contract. And as fluid from the kidneys fills into the bladder. It's urine at this point. As it fills the bladder, we have stretch receptors here that eventually tell us that it's time to release all that fluid. And so at the appropriate time and in place, we relax the muscles of this at the end of the bladder, leading into the urethra. We relax those and allow the urine to flow out of the urethra and out of the body. Again, here's a little more slightly detailed version of this, same structures. You should know these parts, right? We have the kidney, the ureters, the bladder, the urethra, and of course the aorta and the vena cava with the renal arteries and veins go into and out of the kidneys. Now let's take a look at the actual filtering part of the kidneys themselves, because there are millions and millions of these structures embedded within the kidneys, and these are the things that do all of the actual filtration and reclaiming of materials and eventually forming the urine. So these structures are actually called nephrons. Now, this is a very simple view of a nephron. I'll show you a slightly more complicated one in a couple of minutes. But it basically looks like a big, long, winding tube. And eventually we have a whole bunch of these nephrons, and their winding tubes all feed into them, kind of like tributaries in a stream feeding into a river. These are called collecting ducts. This is where eventually the urine is going to collect and then head out the ureters into the bladder. But take a look at this basic structure, and although it's a simplified version of this, it really shows you that the process of the formation of urine is a little more complicated than simply pushing fluid out of the blood and into a collecting tube. If it was that simple, we would simply push blood, where's my cursor? We would push blood out of this little nubby set of capillaries into a collecting tube and then straight out of the body. But remember, of course, that lots of fluid is pushed into the nephrons, but lots more of it is actually eventually reclaimed. And this is why we have this big, long, winding structure, because reclamation or reclaiming of materials, we need to give it lots and lots of opportunities to happen rather than just a straight tube and then out of the body. We have a very long winding tube with lots of chances along the length of it to reclaim things that the body wants. Now, these nephrons are kind of arranged sort of all around the kidney. The kidney, you don't have to know this, but I'll refer to it in the next couple of minutes. So I'll just define these terms. The outer layer, if we were to take a cross section of this kidney, the outer layer is called the renal cortex. And then the inner layer, you can see these slightly different colored structures are called the renal medulla or medullae. And essentially nephrons are arranged with the filtering part, the initial filtering part, all around the renal cortex. So all of those filtering parts of the nephron are arranged on the outermost part of the kidney, and then all of those big long loops descend down into the renal medulla, back out, not back out, but down into the medulla, and then out of the medulla back into the cortex, and then eventually the material or the liquid, which is at this point urine, hits the collecting ducts, which eventually then feed into larger and larger collecting ducts, which eventually become the ureters. Now, here's a slightly more detailed version of the nephron, and again, to give you just a sense of the complexity of it compared to that simplified version I showed you a few minutes ago. And again, what I wanted to impress upon you was that it's super long and convoluted because there's lots and lots and lots of chances along the whole length of the kidney, or sorry, the nephron, to reclaim substances which are initially filtered through. So the four basic steps of the formation of the urine are filtration, reabsorption, a little secretion, and then eventually excretion. And so if we want to work backwards, eventually what we're going to produce is urine. And urine is mostly water and mostly urea, as well as some salts and some metabolites, other substances that we wanted to get rid of. But all of those substances are initially dissolved in blood. And so in order to get them out of the body and into the nephron, or out of the blood and into the nephron, we first have to filter the blood. And it's exactly what that word sounds like. Blood is pushed into a little a cup-shaped structure called a capsule. And as blood is pushed into the small capillaries in that cup or capsule, the force of the blood actually pushes dissolved substances and water out of the blood plasma and into the nephron itself. But it's a very crude process. And by crude, I mean it pushes a whole lot of stuff through. There's not a whole lot of sorting out that happens initially in filtration. We just kind of squeeze the blood, push water into the nephron, and then we need to spend some time kind of sorting out the stuff that actually got pushed through and pulling the stuff we want back into the body. A lot of water is reclaimed, but also some other substances that are initially part of what we call the filtrate. That water with dissolved substances pushed into the nephron is called the filtrate because it's been filtered. It hasn't been sorted. We filtered the blood and small stuff moves out, but larger stuff stays in the blood. We haven't really sorted the stuff out at this point. So then we have to reabsorb it. So there's our reabsorption step. And sometimes we want to actually secrete things into the filtrate that weren't initially part of that filtration process. Really quickly, before we move on to the next more detailed version of these steps, wanted to point out that filtration pushes very, very small things into the nephron. Things that stay behind that are not part of any filtrate or should not be part of any filtrate include large substances like blood cells. We do not want to be peeing blood. We don't want red blood cells to be pushed through. They're far too large to be pushed through into the nephron. And so red blood cells and white blood cells and big things in the blood like platelets should never be squeezed through. They're far too large. But also, interestingly, proteins, not amino acids. Amino acids, they can be pushed through. They're small, but proteins should never be pushed through. And in fact, one test that doctors will do as a test of kidney function to see if you are perhaps suffering early stages of kidney failure is to measure the amount of protein which is in your urine. If there's any protein present, or if there are even trace quantities of protein present, it likely means that the kidney is a little bit leaky and probably some proteins are getting squeezed through when normally they should not be. So big things stay behind. We're really talking about water and dissolved substances that form the filtration or the filtrate in the initial steps. So let's take a look. Again, the overview here is that blood enters the kidney through the renal arteries. A lot of that filtration happens in those capsules that are all part of the outer part of the kidney here, and then eventually the filtered blood returns back through veins to the renal vein and then back into the inferior vena cava and back to the heart. That's the overview. Okay, so let's quickly take a look at filtration. This is a super, super simplified version of a nephron, right? So here's our cup or our capsule. We have a long tube-like structure, and then eventually we have the duct, the collecting ducts, which will collect the urine. And I have the numbers here for the different stages that we just talked about here: filtration, reabsorption, secretion, and excretion. They're numbered so you can follow along. And we'll start with, of course, filtration as the first step in the formation of urine. So what you see here again is a blood vessel. It's come in through the renal artery. It's been branched and branched and branched until it gets down to the very smallest blood vessels that form capillaries. And again, the force of blood being pushed into these capillaries pushes water out of the blood plasma and into this cup-shaped structure. I don't care if you know the name of it. There's a lot of complicated names for structures, but this is called the Bowman's capsule named after presumably Dr. Bowman. And so the filtrate then initially enters this cup or capsule, and then unfiltered blood, again containing lots and lots of large materials like red blood cells and white blood cells and things like that, unfiltered plasma, filtered plasma returns back out through these veins. So again, what we have here then, and let's sort of break this down because this is important. What we have in the filtrate is water, and a lot of it. Remember that statistic, something like 180 liters a day of water with its dissolved substances are pushed into nephrons, but most of that water is going to be reclaimed later on. We also have some salts. Salts dissolve easily and can be pushed through and filtrate. We have some glucose. We have amino acids, but remember, not proteins, and even some drugs or drug metabolites. And here's a list of substances which could be detected in a urine test. These are banned or illicit narcotics that eventually are broken down and excreted through the urine and can be detected that way. So they're definitely part of the filtrate. Here's a little closer view of this process. Again, unfiltered blood comes into this big, nubbly, wobbly looking structure. This is the capillary. Lots of surface area here for blood as it passes through the capillary to be pushed. And all of the filtrate material, or water with all its dissolved substances, enters this Bowman's capsule. And now we have filtrate, right? We filtered it, but we have not yet sorted it. So you guys could probably think about this at home, and you can think about the substances that we would want to excrete, right? Of these things that enter the filtrate, what substances do you want to get rid of? Another way of thinking about this is, of these substances, what are things that you would want to put back into the body, right? So if we're sorting these things, we're going to try and sort out the stuff we want to keep versus the stuff we want to excrete. Well, you could probably guess this, but the things that we want to excrete would definitely be drugs or drug metabolites, right? These are substances that our body has broken down and we need to get rid of. Some water eventually is going to be excreted, and definitely urea, right? That's one of the two main functions of the kidney, is excretory, and we want to get rid of the products of protein digestion. Now, of the things we want to reclaim, we definitely want to reclaim a lot of that water. But we also want to reclaim salts. We want to get some of our glucose back and our amino acids. These are all things that the body can still use. So along the length of the nephron, we are going to be reclaiming the glucose that we excreted, amino acids, salts, and some water. So the reabsorption process, I show it happening along this big long loop. Again, there's a name for this structure if you want to know it. It's called the loop of Henle, H-E-N-L-E. But also realize the reabsorption happens basically along almost the entire length of the nephron. We can reabsorb materials like glucose and water, but I'm going to show it as happening primarily in this big long loop here. But let me quickly skip to the very end of our nephron, right here. And before I actually tell you what's happening right here, I'll point out, this is important, that we're going to be able to reclaim or reabsorb these substances because our nephron is going to be permeable in different places to each of these substances. So we have different parts of our nephron which are permeable to glucose, parts that are permeable to salts, and parts that are permeable to water. And we may actively transport some of these materials out, or we may allow it to happen passively. But the bottom line is, at different places, and I don't think we need to really get down too much into detail here, but different parts of this nephron are permeable to different substances in different places. But let's just take a quick example. This part of the nephron, this is called the distal convoluted tubule. It's very convoluted and it's distal, which means away from that capsule, that initial filtering part. But this part of the nephron is permeable to salts. And we actually can actively transport salts outside the nephron and into the interstitial fluid around it. And all that means is we're basically actively moving salt out. And that, remember, that whole area here, right, is now going to become salty. As the salt's pumped out of the nephron, we're now creating a very salty, and here's a word you should know, we're creating a hypertonic environment. we are actually creating an environment outside the nephron, which is salty, a lot of solutes, and it's hypertonic. And I hope you remember from last semester that when we have a hypertonic environment, water will move passively through osmosis into the area which is more concentrated in solute and less concentrated in water. So essentially, we're creating an environment here which is going to allow water to passively move through osmosis out of the nephron simply because it's a hypertonic environment. So we pump out salt here, water moves out as the filtrate moves through the nephron, water moves out. It's actually very permeable to water at this point in the nephron. And so water is moving out. And in fact, if it's hypertonic enough and if we have the right hormones present, we can even reclaim some water through the collecting duct where we typically think we have the finished product or urine. If it's really salty out here and we have the right hormones present, water can again move actively or passively rather out of the nephron and back into the body. So we've reclaimed two things here. We've reclaimed some salts, and by creating this hypertonic environment outside the nephron, we've reclaimed a lot of water. So the reabsorption process then is happening along the entire length of the nephron. We're sorting out the stuff that was initially part of that filtrate. Now, in some places, now I have number 3 with an arrow pointing right here. This is called the proximal convoluted tubule. Proximal means close to. This was the distal convoluted tubule. Again, I don't care if you know the terms here, but right, this part of the nephron is an area where we are actively transporting substances in. In other words, we have stuff out here, right, that we want to excrete, and it wasn't part of the initial filtrate. So we actually can actively transport these substances in. So for example, some poisons and metabolic byproducts, as well as hydrogen ions and drugs, can be actively transported into what is becoming eventually urine. It's still technically filtrate, but secretion, right? We secrete things into the nephron, which we will eventually excrete. So maybe that those two terms sound similar, but we are secreting into the nephron to help form the urine, which will eventually excrete these substances. Once we have filtered, reabsorbed, and secreted, eventually the now heavily modified filtrate, which is almost now going to become urine, travels down these collecting ducts, which join together eventually to allow the urine to drain out of the kidney. Remember, depending on the conditions in our body, our endocrine system can send hormones which make this part of the nephron, the collecting duct, either permeable or impermeable to water. And that will allow us to either make more concentrated urine, which has less water in it, or more dilute urine, so more water stays in the filtrate and exits the body. So we either can make super, super concentrated, dark colored pee, say you're hot and sweaty and it's been a really hot day outside, and you're trying to save as much water in your body as you can. Well, you don't want as much water to leave the collecting duct as you normally would. So we make this part super permeable, so more water actually moves out of that filtrate, and the urine becomes more concentrated and you make less of it. Or if you drank, say, a whole lot of water, you're not hot and sweaty, but you drank 2 liters of water, your body needs to get rid of that, so it actually makes this less permeable to water. Even though it is hypertonic out here, it's impermeable to water, so more water leaves the filtrate and eventually becomes urine, and so you actually wind up producing a greater volume of urine, which is more dilute. That's it in a nutshell. Again, I would like you to be familiar with some of the basic architecture of the nephrons. I want you to know the 4 basic steps involved in making urine, and that the kidney is an organ which both excretes waste products but also helps to regulate the water content in our blood. I wanted to very quickly just mention that my dogs are barking. I wanted to mention that kidneys can eventually fail under certain conditions. And this can happen through a number of human conditions, which basically put a lot of pressure or excess um, uh, pressure on those little tiny capillaries in the Bowman's capsule. They're very sensitive to blood pressure. And so if you have excessively high hypertension or high blood pressure over long periods of time, that can damage those structures. Diabetes can also damage those structures. Even certain drugs and alcohol can cause those structures to be damaged. And eventually what you wind up doing is not producing any filtrate. And if you don't produce any filtrate, you don't produce any urine, and the kidneys can completely fail. If this happens, you may rely on a machine which helps to filter your blood for you. This is an example of a kidney dialysis machine. Depending on the severity or degree of your kidney failure, you may need to do this a couple of times a week. My own sister, who because of diabetes had complete and total kidney failure, was on a dialysis machine every night. Before bed, she would hook herself up. A simpler version of this, but you can see there's a bag here that eventually fills with urine. Now, these are, to be very clear, these are not nearly as effective as our own body's kidneys. Right? You can live on dialysis for weeks, months, or sometimes years, but they certainly don't do the same thing that our kidneys do as effectively. But essentially, we're taking blood out of the body, pumping it through some filters, which are selectively permeable to certain substances, specifically, of course, urea. We want urea as we push it through these filters, to eventually be filtered out. And then we create this dialysate, it's called, which is basically urine. And then the filtered blood is pushed back or pumped back into the body. And this can take probably 8 hours or 12 hours to complete. Again, depending on the degree or severity of your kidney failure, you may need dialysis a couple of times a week or perhaps every single day. And there's a close-up of it. Now, we're going to talk a little bit later this week about kidney transplants. Again, this is something which is near and dear to my heart. I'll show you the picture. This is a kidney transplant in action. This is years ago, back before I had gray hair. In 2005, I was a kidney donor to my sister. This was taken the day after surgery. Both of us were up and moving around. But kidney transplants essentially replace a non-functioning kidney. In fact, this is interesting. They actually leave the old non-functioning kidneys in, and they simply transplant a new one into your sort of pelvic cavity. So you now have 3 kidneys, 2 of them don't work, but now you have a kidney which does, and you can live with 1 kidney, lots of people do, and that 1 kidney will now take on the role of these 2 non-functioning kidneys. Sometimes finding an appropriate donor can be difficult. You have to find somebody whose immune systems are as similar to the recipients as possible. And so if you wanted to donate, say, to your cousin and you weren't a close match, you could still participate in these things called kidney chains, where, because there's more people who need kidneys than there are donors, we sometimes do these kidney chains where somebody will donate to, let's see if I can look at this. So this person will donate to another person who needs a kidney because he couldn't give it to his own recipient. And the hope is that somebody else that they match them with or paired them with would donate to his recipient, right? So say if I wasn't a suitable donor for my sister, but there was somebody in Arkansas who was an appropriate recipient for me, whose immune system matched mine closely enough, I could donate to them, and then whoever was going to donate to them could donate to my sister. Or it gets a little more complicated with chains of people if we can't find exact matches through a pair of recipients and donors. In any event, this is one way that people are looking at finding more people who can receive live organ transplants or live kidney transplants, and filling the need or the gap between the number of recipients and the number of donors. That's it for the kidneys for now. Again, later this week, we'll talk about maybe the ethics of kidney donation, and we'll do a very simple urinalysis lab. Thanks for listening this far, and I'll see you next class.